My name is Jim Mansfield. I'm the Vice President of Imaging, and I'm going to be walking you through an exciting new technology that we have developed using our Imaging Mass Cytometry system. So first of all, I probably don't need to convince many of you, if you're watching this, why spatial organization matters. It's become increasingly clear that understanding the spatial distribution of the different types of cells that go into a tumor and a tumor microenvironment are really important in understanding what's going on in cancer and, of course, in any other disease, particularly where the immune system is involved. And when you look at the little figure on the right, it's out of a textbook and it's lovely, but real tissue is not that way. It's not all colored with beautifully shaped little cells. In order to visualize that in a tissue section, you need to be able to add markers to that, add in our case, metal label tags on antibodies or RNA to be able to visualize where all of these things are. And in general, that whole area has led to two different fields of technology development: spatial transcriptomics and spatial proteomics. They were each named Method of the Year by Nature Methods, one in 2021, one in 2024. It's interesting that spatial proteomics came later because it started long before spatial transcriptomics. In fact, it started back in the 1940s and really started to develop in the 70s with the development of IHC, immunohistochemistry, and has grown ever since. But in general, up until very, very recently, these have been totally separate technologies, and combining them has been very difficult. So I'm going to talk to you about the way that we do that, and it's using something called Imaging Mass Cytometry. Imaging Mass Cytometry is a multiplexed pathology method, but it does not use fluorescence. Instead of using a camera as a detector, we use a mass spectrometer. So what that means is we put metal-labeled antibodies onto tissue. We then ablate that tissue. We blow it up. We suck the plume into an ICP, an inductively coupled plasma system, which turns it into a plasma, and we separate the light elements, the oxygen, the carbon, the nitrogen, the phosphorus, etc., from all of the heavy metal elements. And we run the metal elements through a time-of-flight mass spectrometer. We measure how much of each metal is present in that pixel, and we raster scan that and build up an image. And then you can do data analysis on this. It's a really interesting technology. It has a lot of advantages that I'm going to talk about at the end of this presentation. But you just should know early on that it is moving closer and closer to the clinic. There is a group in Switzerland, Navignostics, that has gotten the Swiss equivalent of a lab-developed test, an LDT, and they are running this on clinical samples. There was a recent study by Bernd Bodenmiller and others showing that IMC could be integrated into a clinical pathway that was effective and stable and low cost. And one of the reasons that this can be done is because of the stability of the system. So this is the Hyperion XTI imaging system. This is what I'm going to be talking about. It's the original high-plex proteomic imaging system. It was launched in 2017. We're now on our 4th generation, and it can include a slide loader if you want. And I'll be talking later about the easy assay development, the flexible acquisition modes, and the quantitative nature of the data. But what I'm going to talk about first is how to mix that with spatial transcriptomics. Spatial transcriptomics is an excellent technology. People have done some really wonderful studies on it. And spatial proteomics is similarly excellent, but they don't show the same thing. You can tell really clearly from this example. This is a mouse brain. In the area of the substantia nigra, there is a similar but not the same expression between the transcript for the dopamine transporter and the dopamine transporter protein. But in the caudate putamen, it's totally different. The protein shows a lot of that protein, and the RNA shows none of that RNA. And this is probably because that protein was generated some time ago, and the RNA has been reabsorbed into the cell and no longer exists. And so I've got some points here about how they do different things, which is very true. I think it's safe to say that these are synergistic. What I think, though, is important as we grow into the immunology field is that, in general, the definition of what cell types are in immunology, the ontological definition of, for instance, a cytotoxic T cell or a Treg or a TAM, is based on protein. And people have tried to work out ways to infer that from immune cell phenotypes, but that is still just an inference and is not quite always correct. So the two really go well together. Caleb Marlin at Oklahoma Medical Research Foundation developed a really cool methodology that allowed them to take slides that had been run on a 10x genomics Xenium system. They remove the cover slip, as is typical. They do an H&E stain. You know, this is the typical workflow for a Xenium. And then they do an image of that H&E. So now they have the 5K transcript images and the pathology image. And then they remove the cover slip of that same slide, add the imaging mass cytometry metal labeled antibodies to that, and then we image that on the Hyperion system. So this gives 40 or more proteins detected by IMC of the exact same slide that was run in the Xenium. So this is a very flexible technology. It can follow many other modalities, not just spatial transcriptomics. It works on an H&E. It works on immunofluorescence, both cyclic and regular. It works on spatial transcriptomics. And to be honest, we're just trying to figure out which all systems can it be validated on. Every one that we've tried so far has worked. So I'm going to show you some examples of this. Here are some images. The one on the left is the imaging mass cytometry image of this sample, showing the DNA, the nuclei in red, the keratin in green, and CD45 in red. And the image on the right is also run on an IMC system, but it's run after the Xenium process. So the one on the left is without the Xenium process, the one on the right is with, and they look incredibly similar. There are some differences in intensity, which is probably to be expected because there's a protease step in the Xenium. But for the most part, it's quite good. And I would say that probably 80 to 90% of the markers that we have tested have been working. So it's a really good transfer of being able to do full spatial transcriptomics and then full proteomics on the sample afterwards. Here's another example, three different immune cell types. The one on the left, again, is only IMC. The one on the right is IMC following Xenium. And again, they look quite similar. None of the markers have been lost. We, like 10x Genomics, have our own cell membrane or cell segmentation kit. So just to show you that in our system, the cell segmentation kit persists, will work well after, whether it's with a regular IMC or IMC after the Xenium. And so that helps a lot with cell segmentation issues down the line. This kit involves 3 different proteins that we label with metals. And to show you a little bit about how they can go together, so there's a number of third-party softwares that can be used to merge the protein images along with the transcript data. The transcript data, of course, is not really an image. They save it as a big text file, the location of every transcript spot. But they can be merged and they can be analyzed. And I'll show you a little bit more about that in a second. But I think these 3 are a really good example of the correlation and non-correlation of these type of markers. The leftmost image is keratin in red and the transcript for keratin in green, and so they mostly correlate. There's 1 or 2 areas where they don't. The middle one is EPCAM, the protein for EPCAM in red, the transcript in green. They correlate very well. But the one on the right, beta-catenin in red, and TCF7, the transcript for beta-catenin in blue, and you can see they almost anti-correlate. There's a lot of places where the beta-catenin is strong and the transcript is weak. But because we can do excellent alignment and co-registration, this allows you to get per cell data on both protein and RNA. So here's an example of that. In this image, the white, the grayscale image is the nuclei from Imaging Mass Cytometry. The red outlines are the nuclear segmentation from Xenium Explorer. And if I blow that up, you can see that it's actually really good correlation between the two. Per cell, because it's the exact same slide, it's easy per cell alignment. And if you're not the kind of person who likes to go on GitHub and download programs, or if you don't like Xenium Explorer, there is a company called Aspect Analytics that makes a software platform called Weave, in which you can merge and align and analyze all three of the images that are being done here: the IMC image, the 40-plex protein, the RNA data, the 5K transcript data, and the H&E image can all be aligned and merged and overlaid. And then you can take that and do per cell analysis, so per cell phenotyping on protein and RNA simultaneously. It's a really interesting technology. And I'm going to tell you a little bit more about why imaging mass cytometry is a good way to do this. So this is the Hyperion XTI imaging system. You saw this slide before. I'm going to talk now about the three key points of this: the easy assay development, which I think is key to this particular experiment, the flexible acquisition modes, and the quantitative nature of the data. Because imaging mass cytometry uses metal-labeled primary antibodies, it makes it really easy to develop your assay. Once you have an antibody with a metal label on it that you know works, that you've figured out the titration for, you can mix and match that with any other technologies, any other metals and antibodies that you want. We have over 300 labeled antibodies right now that are validated. We have them in panels, we have them individually. You can easily swap markers in and out. But what's probably really important for all this is there's a simple custom antibody labeling step. So if you have a clone that's particularly important to your experiment, you can just label that with a metal, throw it in your panel. It's a one-step staining and one-step imaging, so you don't have to waste precious samples post-Xenium to validate whether this works or not. Here's an example of some of the modularity of our panels. We have a basic human immuno-oncology panel made up of 31 markers, but even that is made up of subpanels. You can put all of them in, you can leave a subpanel out, and then we have a bunch of other panels that can be mixed and matched with this. And of course, optional markers of interest, custom clones, individual markers from other panels can all be swapped in and out in a really simple pathway. That is very different from how things work in a cyclic system. I think the imaging modes really separate how Hyperion works compared to other proteomic imaging systems. Because we do a raster scan of metal-labeled antibodies, we have developed three different modes of imaging that can help you get information out of your sample. There's tissue mode, so this gives you large 5 by 5 micron pixels. These are still subcellular, but not really cellular resolution. A typical cell is 10 to 20 nanometers, so it's below the size of a cell, but not really enough to visualize individual cells. But it allows you to image the entire tissue very quickly. We also have two modes that go together. The preview mode, which is a 1 micron resolution, so a small pixel, but we take one every 25 microns. So this gives a really low resolution image of what you're looking at and allows you, but it's all 40 or 42 markers at the same time. So this allows you to decide which areas do I want to go in and get high resolution images at 1 micron resolution. We do also have 0.3 and 0.5 micron resolution available as well. The nice thing about these modes is you can mix and match them in different ways to really increase your throughput. You can get up to 100 slides per week with cell mode, with cellular resolution in parts of your sample. Or you can do 100 slides a week at the 5 micron resolution. Or if you really want, you can spend a lot more time on one slide and get a lot of high resolution cell mode data. When you're looking at combining Hyperion with any of the other technologies that are on the market, you can, in theory, you can mix the Hyperion with any of these technologies, although we have not yet tested the cosmics, but all the others work. You can see that the Hyperion has the largest imaging area. So whatever area you have imaged on your other system, you are going to also be able to image on the Hyperion. One last mention now about quantitation. We don't use a camera, and so we are not limited by all of the camera optics and resolution requirements and the NA of the objective and the dynamic range of a scientific CMOS detector. We use a mass spectrometer, and so this means that our linear dynamic range is much, much higher than any of the other technologies. You're lucky if you get 3 orders of magnitude linear dynamic range in fluorescence. It's probably less than that, especially if you've amplified your signal in any way, that reduces your linear dynamic range. So we have a huge dynamic range. And on top of that, we have extremely good limits of detection. We can detect down to 100 antibodies per cell. So that's 100 copies of an antibody in a cell. And that is just above the noise level. That's, of course, the 3-to-1 signal to noise for detection. But as you can see from this example, we can, because we're using metals, we can actually create a calibration slide. You can see that we are linear for 5 orders of magnitude dynamic range, and that we can get right down to the detection limit of 100 antibodies per cell. This is not possible with any other technology. So let's look at an example of that and why this is important. Like, okay, so you have high linear dynamic range and you have good quantitation. What does that mean? Well, here's an example of a tumor that we were sent that's a HER2, a breast cancer tumor. And the image on the right is what we were sent for the pathology. This is the immunohistochemistry, the DAB stain of that sample. But it turns out, if you look at the fluorescence light, the color image on the left, or the simulated bright field IMC image in the middle, there's 2 different parts to this tumor, one with a high expression of HER2, one with a low expression. And so we can capture that. And this can play out in a lot of other ways. So here's that same tumor. We use 2 different HER2 clones, and the interior domain and the exterior domain. And so you can see that the ratio is slightly different, which is interesting in itself. But mostly you can see that there's areas of low HER2 and areas of high HER2. And if we look at that now with the other 40 markers that were in this sample, if in this case, the low HER2 is the top and the high HER2 is the bottom, you can see that the Wnt signaling from our metabolism panel is very different in this tumor. In the low HER2 area, the Wnt signaling is on. In the high HER2 area, the Wnt signaling is off. And you can look at those more closely and see the differences at high resolution in the cells. And you can keep going with this. We can show different aspects of the citrate cycle, the CPT1A, fatty acid oxidizing enzyme, etc. These all correlate to some degree with the low and high HER2, although in the case of the fatty acid, not entirely, which is interesting in its own right. And so it's important to know for a lot of these markers that you can quantitate both low weakly expressing cells in the same sample as strongly expressing cells, or in the same image as strongly expressing cells. So here's 4 different examples. These are all really important markers for quantitation. And because of our high linear dynamic range and the fact that we don't use amplification means that we can image them correctly. And so just one last thing, this is a preview of our metabolism and signaling panels. There's a whole talk that goes with this. I've mentioned these as I've gone, but the panels are really interesting to anybody looking at cancer metabolism or signaling, and they work really well and, of course, give beautiful images. So with that, I would like to just conclude by saying Standard BioTools has a lot of different technologies. We are developing new things all the time. And of course, spatial biology is hot. Spatial proteomics is a big part of that. The Hyperion XTI is a really good fit with spatial transcriptomics. It has a lot of advantages. And the new application areas in spatial biology, oncology, neuroscience, HER2 low and other low weakly expressing markers are really important. But the point of this talk was to tell you about the multi-omics with spatial transcriptomic imaging. And so hopefully you have learned something from this. Thank you very much.